Good day, my dear postgraduate students. In the last class, I had given you a question to identify the structure and its relevance in pathology. Also, I had given you one other question. What is reverse polarity? There are a lot of labels over here. Let us see what is in store for us. Welcome to Glide the Slide 17. The organism here is the leishmaniasis and the disease is Kala-Azar. This is the sandfly that transmits the disease, more common in Africa. There are basically two forms of it. One is a pro-mastigote form and another is a mastigote or a flagellated form. This is the life cycle, and the human being happens to be an intermediate host, just as in malaria. We can start anywhere and then see what is in store for us. So the sandfly, it takes a meal and then it regurgitates. This is important. You find that after the meal, there is a regurgitation and it is stored in the salivary gland. There has to be an expulsion of the saliva so that it can aspirate blood. In the process, you find the parasite is being inserted into the new host. You find that there are the pro-mastigote forms which develop. And the pro-mastigotes develop into the amastigote form. The amastigote form is something like a cyst. It is not able to proceed much, but then when the situation is congenial, it can be converted into a full-blown parasite. That is a basic principle in parasitology. So here, it takes another meal and you find that the amastigotes are converted into the pro-mastigote form. I hope things are very clear for you people. There is the amastigote form, then there is a division, the sandfly. It takes the meal, it is being injected by means of the proboscis. And in the human being, it is getting converted into the mastigote form or the amastigote form. After which, again, there is a second injection. It goes in for the completion of the cycle. This will be identical in the case of malaria also. What are the signs and symptoms of this disease? In the early case, it can be just malaria, fever. Maybe there can be a fever with rigor also. Fatigue and weight loss. Very non-specific. Hematologically, there can be an anemia and a leukopenia. Obvious reason can be a bone marrow infiltration leading to some kind of an aplasia, resulting in a thrombocytopenia as well. Hypergamma globulinemia because of increased activation of the liver. There is a hepatomegaly and a splenomegaly. Cachexia, hepatic dysfunction, and there can be a dark skin. Look at the skin. The spleen over here, it is almost trying to cross the umbilicus. So in some conditions where there is a massive splenomegaly, the spleen may be palpated in the right iliac fossa by the edges. Now this is a histological picture. Obviously, I am able to see a very pale outline, which means that they are all the foamy histiocytes over here. And in this, I am not able to, though it is, since I am already prejudiced with the answer, I know a little bit, but it is difficult for you people. Try to concentrate. You find that there are some cells. You find that there is something like a collar button that I'm seeing over here, and another one. These are all too small for the nucleus of any other cell. So these are the parasites which are seen. courtesy pathology outlines. And this is the visceral leishmaniasis. So obviously it can be a lymphoreticular one. I am able to see the lymphocytes, plasma cells, and the histiocytes. Now look at this. Within a histiocyte, I am able to see this multiple dot and dash forms. These are called the amastigote forms. Look at this, very clear. In a still higher magnification, I am able to see a dot and a dash. What is this dot and dash? We shall see. Again, very beautifully made. This was one of my exam slides, and we are able to diagnose it. And in some cases, the cell might be ruptured, dispersing the amastigote forms to the periphery. But all said and done, the dot and dash are very clear for us to diagnose. Thanks to Lancet for this picture. Look at this. This is a flagellum. And as I told you, there can be something like an axoneme. Axoneme usually you do not see. There is a basal body which is ovoid and a kinetoplast which is perpendicular to it. This produces the dot and dash format. The rest of the cell is not seen. The nucleus is there, the cytoplasm is granular. This is the amastigote form of the leishmania. That is what you people are seeing. It is again a repetition. Blepharoplast and then this. I chose this particular one because only earlier we had seen a slide of ependymoma in which there will be the blepharoplast which will be away from the apex of the cell. That is why you get something called reverse polarity. So I hope you people are able to make out the cells which are infiltrated with this parasite. There is a hepatomegaly and a massive splenomegaly also, according to the MRI. So this is the picture that we had seen earlier. I hope the dot and dash is very clear for you. The history is important. A fever with rigor, a patient somewhere from the band of Punjab to Bihar, bone marrow aspiration, and the inclusions are seen both within the cell and outside the cell. This is about the amastigote form. This is what we are trying to tell you people. It is rounded 2 to 4 microns. There is no flagellum that is seen. And the whole thing is embedded in a cytoplasm. The nucleus, parabasal body, and the blepharoplast. This is called the LD body or the Leishman-Donovan bodies. Nucleus is eccentric. Whereas this is the promastigote form. The kinetoplast is seen and it is dot-like. The axoneme is there and it develops into a delicate filament which can be the flagellum. The stains which can be used are the Promnowski stains, such as the James or the R.A.T.S stain. And the cytoplasm is pale blue in color, whereas the nucleus is red in color. We shall glide on to the next slide. What you people are seeing over here is a 2-year-old child with abdominomegaly. Identify, describe, and write a short note. Thanks to R.A.T.S for this particular picture. And my dear students, this is the next slide for glide the slide. A 2-year-old child was brought with abdominomegaly. So I am little vague in this, but still, you are supposed to identify the lesion, describe, and write a short note on it. Thank you.